Hi, this is the Bradford assay demonstration video where I'll be basically explaining what we did in our lab and during our experiment. So to start off, me and my lab partner were tasked with testing our actual GFP samples as well as unpurified lysate. In this clip, we've already done our dilutions, which I'll get back to, and we're loading our Bradford reagent first before adding our dilution samples. The purpose behind the Bradford reagent is to bind to whatever protein is in the sample and give us a quantitative measurement of the protein in each of our samples through OD readings on the spectrophotometer. From that point, we can compare these sample readings to previously made standards provided by other groups in our class. Now in this clip, I'm showing off 6 sample tubes we created for our dilutions. On the screen are specific dilutions we created. We tested 2 tubes of unpurified lysate and 4 tubes of purified GFP. Our aliquot was either unpurified lysate or purified GFP, and our diluent is TE buffer. Now that we have our standards and our Bradford reagent loaded into our 96 well plate, it's time we add them together. In this clip, we've added our controls, which are the bovine serum albumin standard for the positive control and the TE buffer for the negative control. Here, we actually made a mistake by adding our 6th solution tube in as our positive control, which should never have been done. If the sample is a part of the experiment itself, it should never be a part of the control group since there are, in this case, unknown factors in that sample. One more thing I'd like to add is we're running 2 copies of the same samples side by side to ensure consistency in our readings. In the clip, we have 2 columns of wells with the Bradford reagent, and each row next to each other are just doubles. So now that we've eventually loaded all of our samples, each in volumes of 5 microliters of the GFP sample and 250 microliters of the Bradford reagent in each well, we can run our entire plate in the spectrophotometer at 595 nanometers. These OD readings will be averaged and compared to known standards. After 10 minutes of incubation, this is what our well plate looked like before readings. And this was one of the higher standards from another group after 10 minutes. Thank you for watching, and I hope this video demonstration was helpful.